Now the plague count in the bacteriophage titrations. Plague count is just similar to the colony counter, which is used as the bacteria. We see it in the detail. The plague values, the plague counters is plague is the counts analogous to the colony counter as for the bacteria, and then the values which are present will be directly proportional just the number of the viruses or just the number of the phages which are isolated on some specific kind of the plates. Just like you can see here, if we take the example, the phase 1 titration, you can see here the 3 different kind of the plates, and it will be just like the colony counter in the bacteria. Here you can see the phages are appeared just like the colonies in the bacteria, and then its values are proportional to the amount of the phages plated. If you are plated it just like this, then the values will be directly proportional just like we have plated it on the specific kind of the plates. The plate cultures are prepared with the combining or mixing of the phage mixing with the top agar. The top agar is very important. You have to use it for the centrifuge. First, the agar preparation is very important. You have to centrifuge and then you have to put it at 37 degrees centigrade before it is going to be used. Because when the procedure of the phage mixing is the top agar is the viscous material, so when at the time of the vortexing there will be some kind of the air bubbles which will be produced and which should be avoided, or which should be reduced reduced as much as possible, so that we can see it how much is the phages are present. So top agar preparations contain lower concentrations of the agar, and then it may be about 7 gram per liter, and then the normal solution used to prepare the agar plates, it will be about the 15 gram per liter of the agar which can be used. Here you can see that graph, the presentation of the agar, and how you will interpret the results with these bacteriophage titrations. On one side is the plague count and the other side is the relative concentration of the phage. So it will be, the results will be shown in the form of the straight line, but on the here that how much relative concentration of the phage, and then similarly the plague count. We can also see in another slide and the procedures, about the procedure of the phage titration of the phage count. Here is the phage stock, then the plaques formed on the lawn of the host. Here you have shown use the plates, and then the phages were collected and cultured with the host too. Here this one, then the plaques formed on the lawn of the host 2, which be in this way. Similarly, the phage collected and cultured with the host 3, and then plaques formed on the lawn of the host 3, and then you can see that from the different hosts you have collected the bacteriophages, and you need to see it in the laboratory. Here you can see the phage bacteriophage titrations. The lower agar concentrations allow the progeny phage from the lyse cell to diffuse through the media and infect the neighboring cells. Of course, here we have taken 2 kinds of the examples. One is the CRISPR and the other one is the anti-CRISPR. High virus and the viral replication, the reinfection of the entry, and then it is the 2 examples. The low virus, the reinfection is rare because of the viruses, and however here is a reinfection. Then after the lysis, the plaque, the zone of the lysis is produced on the plates. Now the plaque's size, which is very important, depends on the how soon the bacteria in the agar reach the stationary phase and stop the reproducing. Then the plaques will stop and spreading at this point, and then they will see that how much is the bacterial titrations has been occurred.